So you can start now, sir.
Oh, okay. So can you please tell me if my slides are visible? Because I'm sharing it.
Yes, sir, it is visible. Just make it full screen, sir.
Yeah, sure. I'll do that. Okay. Yeah.
Perfect.
Okay. Yeah. So welcome you all. I'm Sangram Bagh. I'm a associate professor at Saha Institute of Nuclear Physics in Biophysics and Structural Genomic Division. And what I'm going to talk about the synthetic biology and the space bioengineering. So today is like an introductory class where I'll just try to give you an introduction of the synthetic biology and its general applicability. So you may heard this term synthetic biology and you may not heard, but synthetic biology is kind of a new subdiscipline of a biological engineering or the biotechnology or genetic engineering in what way you are going to tell. But there is a very fundamental difference between a conventional biotechnology or genetic engineering, the synthetic biology. So if you see in my slide, I mean in the title slide, there is a, it looks like a Lego block somewhere in the middle. So when you talk about engineering, let's say you are thinking about the computer you are using for this class at your home or at your office or at your class, wherever it is. So those computers, there's a lot of modules are there. Like there's a RAMs there, there's a motherboard and something. And if anything actually gone bad, like for example, your memory, the RAM, then you actually take it out and put something new RAM, or you can increase your RAM. So it looks like that you can actually assemble many of the parts to make your computer. Okay, so that is the one of the beauty of the engineering, that there's modular and you can connect on a particular design to develop a new system or new functions or whatever you say. Now, in things of the biological engineering, it's not like the case. So if you want to develop some genetic engineering, like you heard, like, for example, if you want to genetically engineer a rice and try to make, put some beta carotene there, you probably heard about the golden rice, which has been developed in India, which has the beta carotene there. So that rice, if you can take that, so you are getting the beta carotene, there's a vitamin A with that. Now, making those kind of things, very difficult. It takes like more than probably 10 years, and there's a lot of trial and error. You do not know how to do that, right, at the very beginning. But you imagine a kind of the biological engineering, which is just like a Lego blocks, and you have all those modules, and the way you actually develop an aeroplane or a house, a two-storied house, a three-storied house, a kind of a garden or something using your Lego. Imagine one day the biological engineering is something like that, when you have those kind of modules and you join and actually develop a new function or new cell, or it's a new organism in that way. So that is the fundamental philosophy of the synthetic biology. And what we'll examine through this lecture, that where are we and how it's, I mean, one day how you can go to that. What could be the principles that way? So that is the that's the motto of the synthetic biology. But before going to the synthetic biology, I'll just talk something about some other technology. Okay, now you probably actually have seen this movie, which is called Jurassic Park, or any of its sequel. Probably you have actually watched that now. So this is based on a novel called Jurassic Park, written by Michael Crichton. Okay, and it is in 1986. So in this 1986, definitely it's in the science fiction. And what we're going to test first, that so whatever the technology we have today in 2021, can you make a dinosaur? Or so the very fundamental question is how to make a dinosaur. Okay, so very first thing, if you want to make a dinosaur, is you need the blueprint of the dinosaur's life. That is its whole genome sequence. Okay, so in an actual the novel in the Jurassic Park, you probably have seen, and also in the movie, that actually a company, they somehow dug out some of the embers, and basically there's a mosquito which had some dinosaurs bloods in it, and it is actually preserved in the amber, and it fossilized. So after millions of the years, uh, um, somebody actually found that kind of the fossil. They actually took that blood, it was preserved in such a way, and from there they actually got the dinosaur DNA. The very first step, that you have to read all the blueprint of this dinosaur, the whole genome of this dinosaur. Okay, and 1986, that was kind of a science fiction, because that time it's only available sequencing technology is the Sanger sequencing, where it is very accurate method, but it is low throughput. And now, if you, I suggest, or I request you to actually read that novel who are interested in the biological engineering, that Jurassic Park. That's a very exciting novel. And the very first thing that their opponent of this company called InGen, they started thinking that InGen is doing something, because they found in the story that this InGen, that company, and they started buying or developing a lot of automatic genome sequencer. And that time, the automatic genome sequencer was not available at all. That's a science fiction story. But today, what we do know, that we can, if we have a genome of the dinosaur, then we can actually read all the genome or all the blueprint of the life by letter, word by word. Okay, and we know that's in 2003, the Human Genome Project has been done, and that's like it's a multi-billion dollars project that time. Today it's a very cheaper at this moment. But what you can see that in 2003, we actually had the technology through which we can actually, if we have the dinosaur DNA, we can actually read the whole dinosaur DNA through that, right? So there is the first part of this technology we have already achieved, which was written in 1986. The second part, that if we get that DNA, how we'll actually synthesize, how we make this whole genome, right? And this is something what we call sometimes it's called artificial life. It is first published in Science in 2008 it is done by Craig Venter's group. And what they have developed, they have chemically synthesized actually the whole genome of a bacteria. It's a Mycoplasma genitalium, and it's you can see it's 582 kilobase per. Now, what is the significance? Because each something says that you can write anything you want. This is completely different than what you know as the polymerase chain reaction. For the polymerase chain reaction, it's like a photocopying, right? Because for polymerase chain reaction, you need a template, and then you actually start multiplying it, like copying it like a photocopier. Whereas synthesizing a genome means you can write anything you want, right? So that's a very important technology, and which when you have this whole genome, you need to write, you need to get that whole genome, and the way you can do that, you can actually write the whole genome sequence by chemical, you can chemically synthesize that thing. So that probably the second part of this technology we have achieved by now. And third part, what I can say, that is the same group who actually published another science paper in 2010, but they have done very interesting thing. And again, they actually synthesized a whole genome of mycoplasma, of a particular species of the mycoplasma. Then that synthesized genome, they actually placed into the other mycoplasma species. Okay, from there they actually took that genome out for the second species and put the genome of the first species. Interestingly, what you see now, that the second species turned into the first species. So we are transforming one species to another just by, and you remember that this is a totally chemically synthesized genome you are putting to another species. So if you see what is the significance, I'd say that the software is creating its own hardware. What does that mean? Because the software, because this is a whole genome, which has the whole information and the programs in it, that is definitely the software. And it is creating its own hardware means that the whole cell, when you're changing from one species to another, because the difference of these two species in terms of the composition of the cell wall, it is the composition, the kinds of the metabolites, kind of the energy uses, and so on, so on. So now when the first species is changing to its second species, what it is doing, it is actually creating its own hardware, that is its own cell, so that it will work. So the significance is that like software is creating its own hardware. So if you see what, I mean, if you want to get a feel that what means in a machine world, it looks something like that. Probably you have seen this movie, probably you haven't. It doesn't matter, but it's a very popular movie, old movie, is The Matrix. And what you can see here in the my right hand side, this particular person, it's not a human, it's a program. He called himself as architect of the matrix. And what, so a program is making another the programs of the matrix. Okay, so the software is creating software. That's always happens. You need that's that happens. But what is important here, the software is not only creating other softwares or other programs, they also create the real machines. Okay, so this is the real machines they're creating that hardware, and this real machine's job is to actually destroy the human settlement in that movie. That's called the last human settlement. It's called Zion. So what you look at, it looks like a science fiction in the machine worlds. In biology, we have actually achieved the way that the software is creating its own hardware. So with that, if you now come to, it's called the Mr. DNA video from the Jurassic Park. Okay, and what this Jurassic, I mean, if you have seen this movie, so this Mr. DNA video is going to talk about how they have actually developed this dinosaur. So that the way they have developed, the first, as I told you, that they got the dinosaur DNA and they sequenced it, the full sequence, and then from the sequence they actually synthesized, okay, the whole genome, and then they took a frog cell, embryonic cell of the frog, and then take the frog's own nucleus out, or the DNA out from this nucleus, and then they poured this dinosaur's DNA in this frog's embryo, and that way they got the dinosaur. So now, but what I, so that is in 1986 science fiction novel. And today I told that we have the technology to actually read the whole genome, to write the whole genome, and to transform that writing genome from one cell to the another. And that gives you the whole technology for making the dinosaur. What we do not have, the dinosaur DNA at this moment, but we have achieved up to that level. Now, this gives a question. The question is, okay, now we can read and we can write, but do you understand? What do you mean? Because in this movie, if you see this velociraptor, okay, which is trying to attack this frightened boy, it's a human and it's a dinosaur. And if I give you the DNA sequence of both of them, okay, just looking at the DNA sequence, can you tell which one is human and which one is a dinosaur? Or even if I write that this DNA sequence is for the human and this DNA sequence is for the dinosaur, can you tell that what from this, I mean, in this sequence, what makes it a human and what makes it a dinosaur? What makes that human on whatever we are doing? It's starting. I mean, at this moment we are making the movie. We're talking about some synthetic biology and everything that is in our brain, and the all things it happened. It's somehow it's written. That information is somehow is embedded in our genome. But looking at this, the whole genome, just the sequence of a dinosaur, or forget about the dinosaur. Just take a tree. Let's say banana tree. Okay. I mean, what else is the what makes the difference between this genome if we give it the sequence? How can you tell that this genome is going to make a banana tree and this genome is going to be a human? So that means that we can read and we can write, but we do not understand at all. Okay, and that is, it has a lot of significance in today's biological engineering, and we'll see what it is. So here, what you can see basically is some of the engineering marvel. So here, when you think of the engineering, this is some building on Singapore, very nice building. And here, what you can see is a rover on the Mars. Okay, so it's a rover on the Mars and things. That's we always think about this engineering. But have we ever seen engineering like that? A Pegasus or a HAL? So that I'm sure that you haven't seen this kind of engineering. So the question is that at this technological era, when you can actually control the movement of the rover on the Mars from the Earth with almost a mathematical precision, why we actually cannot make a Pegasus or a Hawk? Forget about making it. Even we cannot think. There is no way that we can think what is the starting point. So what kind of the changes in the genes and something we need to do in order to start growing some wings on the horse? We cannot even think about that. Similarly, we do not know what kind of the mutation or what kind of the genetic changes are required to make a hawk. So we, even just by thinking, we have no idea, right? So the question is why? So there's a one simple answer. If you think in this engineering objects, engineering things, then we do know for them they have a very well-designed, well-designed design principles of making them. Based on this design, you actually fabricate them, you assemble them, you made them. You test them. If that doesn't work, then again you go to the design, you change it, and you fabricate it in such a way. But we do not know what is the design principle of life. And that actually makes us, it is impossible for us to even think about how to develop this kind of things, okay? Now, so to answer this question a little bit deeper, so we have to look a little bit of the history. And if you in my left-hand side, what you can see actually, Professor Max Delbruck, who got the Nobel Prize in Physiology and Medicine in 1969, okay? And so he, what he discovered is what is the molecular mechanism of the viral replication process. But what interesting is that Max Delbruck was not a biologist by training. He was a gifted physicist. He was a student of Niels Bohr. Okay, and that time, you imagine the physics is extremely exciting. Why? Because at one hand, there is a theory of relativity, and one hand, there is a quantum mechanics. It just came around the corner. And every physicist was so excited because this is something new physics is coming. That means that every physics want to develop some new physics laws. And Max was not also an exception. So he also wanted to get develop or discover some new physical laws. So what he thought, that everybody is looking at the chemical or the physical and the chemical worlds to find actually new laws of physics. So he thought that nobody's looking at the biological system. So he wanted to go to the biological system and try to understand some of the physical laws, new physical laws out of that. And he started working on this biology. And as a physicist, he started from a molecular point of view. Okay. And interestingly, what he found, that he couldn't find any new rule of the physics from the biology. Instead, what he found, that all rules of the physics and chemistry are actually maintained in the biology. Okay, so that's a very important point. At the very same time, it is not just the Max Delbruck, but if you think about Schrödinger and Linus Pauling, they're also gifted physicists, and they also started understanding the biology from a physics point of view, and that is probably the starting point of the molecular biology. But when you were when physicists started studying the biology, they are not only just looking at a different view, but they're also bringing the philosophy of the physics into the biology. And what it is, and this is a reductionism. So reductionism is, probably you know reductionism very well, but just to give you an example. So when you think about your high school physics numerical problems, then you want to do some mechanics, and you want to for a ball or a car or the earth, and in each of the cases you actually imagine a point mass m, right, or m1 m2 or something. Then you imagine this point mass, you calculate, you do some of this mechanical calculations, okay, with Hamiltonian, you solve this and things, and then again, then in your answer it's going to tell that's okay, this car will go up to this path this way, this ball is going to that path that way, the earth is going to, in this time it will go that much of the path. So you reduce everything in a point mass. But in reality, we know there is a little difference. Okay, for example, if you develop a knife, okay, let's say it is a 50-gram knife made of titanium, okay, very sharp and thing. And you have, on the other hand, you have a 50 gram of soft ball, Okay. Now you throw these two things towards a person from a same speed, from the same space, and both have this 50-gram mass. Now you know the way it will hit the softball, that 50-gram softball, the way it hits that person and what is the effect is completely different the effect from this titanium 50-gram knife. Okay, because we know that. But if you want to just calculate it, you reduce them this 50-gram, it was a particular 50-gram mass as a point mass, and you calculate, and it looks in from the your solution that the effect would be the same, but it is not. Okay, so this reductionism has been used in the biology for the last 50 to 70 years. And that is the manifestation. That is why there is a lot of things actually come to the biology. In starting from the molecular biology, now you look for a protein or particularly a gene which is related to a disease. And what is one of the highest manifestation of this reductionism is Human Genome Project. Now we can see there is the president, Bill Clinton, that time actually announcing the success of this, because it is billions of the dollars money, US, and there is other countries from the European Union, Japan, they actually spent. There's a lot of scientists, more than 1,000 scientists they're connected, and it's kind of an industrial scale operation that time. Okay, the whole idea of this big science in the biology was that if you know each and every gene of the human, then we can actually cure every disease, including the cancer. Okay, and that is in 2003, and today is 2021. Definitely due to this genome sequencing, as a human, we have actually there's a lot of, it helps a lot of our medical sciences, but definitely not the way we thought. So we thought that if we know this information of the all gene, then we actually cure the cancer or something, but we know that we could not. So the whole question is, again, that even knowing each and every gene, why we cannot understand the cancer or something? We could not find a specific marker for cancer. What is the reason for that? And again, that's actually embedded in the complexity of the biological systems. Now here is just an analogy that how the biological complexity and some other complexity combines. So here is the network of the computers on my left-hand side. In 2009, world's all computers which are connected to the internet, it's a map of that. Okay, there's a very famous map. And my right-hand side, it is a kind of simplified representation of a biomolecular network of a bacteria. Right? So now just looking at these two things, you see in terms of the complexity, they looks like, in terms of the nodes and these connections, they looks like same. Okay, and at the same time, the way they're connected in the biological system, they're nonlinear. So there's a lot of feedbacks and there's a lot of noise. And when it's a nonlinear, means that is if you want to mathematically you want to see how it is using the nonlinear differential equation and solving it. It is extremely difficult in this scenario. Okay, so the whole point is that a biological function does not depend on a single gene or protein, but a biological function depends on this networks of thousands of the genes and the proteins. And that is one of the reason we failed to actually look at the cancers and other diseases in that way. And that is why we couldn't. So, and this things is so complex, there is no mathematics which can decipher this kind of complexity. That's why we do not know what is the design principle of life, and that's why probably we cannot make a Pegasus or a Hulk using our biological engineering tools. Now, so end of this last century, there's a scientists started thinking, or actually they thought that this biological network, if you would like to understand this network of the genes and proteins, so who are working on this network? So if you see in the engineering, the electrical engineers and the electronics engineers, they're always working on the networks. So why not that we take some of the rules from them and try to understand the biological networks from that. And the people found that, okay, in many ways, a part of the biological networks can be actually explained from this electrical or electronics point of view. And in one way, this is loosely defined as a molecular systems biology. Now, if this is correct, then if this philosophy is correct, then it is also correct that that you can actually develop a small biological networks following the same kind of the principle, and then you plug in to the cell, and you can actually develop a new function. And that is the motto of that's the way of the synthetic biology thinks. So in one way, if you think about the hierarchical principle of a computer, so we have some physical layers, right? So, and some semiconductors and stuff, and through which you are making some of the gates, some of the logic gates, AND gate, NOR gates, and something. And when you actually connect the gates, you make a very integrated device or circuits in terms of the modules. You actually connect those modules, you get the computer, and then you connect the computers to the internet, you get a network. Now, in biology, this is also possible because you have the protein and genes. They're connected to give some biochemical reactions. And that biochemical reaction goes to the pathway. That goes to some of the cells of the tissues, and then go to tissues and the whole organism. However, we are not very sure about the evolutionary principle or evolutionary rules which actually makes from this hierarchical protein to genes and biochemical to pathways. These rules we do not know. But whatever, when we do not know, we take some of the rules, this hierarchical principle, and then just put it here, which basically means here, using some genes and proteins, we like to develop some biological gates through the biochemical reactions. And then through this, by the way, we know the rules from the gate to modules. So why not using these biochemical reactions? I mean, the gates, what we made through the biochemical reaction, we made the pathway, which will work as a integrated device or a modules similar like to this, and then they connect. So this way, probably we can develop some of the new function and from a very design-based perspective. And that is the fundamental motto of the synthetic biology at this moment. So what I'm trying to tell you that you want to develop some engineering principle, let's say in terms of the computation, and you want to put it here in such exactly the same way so that you can actually develop a completely new system. Now, when I talk about this computation, which means you are trying to tell that we are going to develop a lot of complex computation using this kind of the protein, genes, and the biochemical pathways. But do that biological system compute? And we'll see that. So this is a very interesting picture. This is a game of a chess between a computer, which his name is Deep Blue, made by IBM, and Garry Kasparov. Garry Kasparov is like in Vishwanath Anand, and he was one of the greatest chess player in the world. That's probably one of the greatest chess player of the all time. Garry Kasparov, you can tell, and it's in 1997. And so they played the computer and the Garry Kasparov, and you can see what is the results. I mean, I don't have to tell. You can see how Garry Kasparov looks like. So a computer defeated Garry Kasparov. So what it's defeated? It defeated the brain of Garry Kasparov. So we all know that brain actually computes. So whatever, so at least we believe that all our intelligence, the consciousness, and everything is coming out of the brain. And we do not know much about the brain. We know a lot of its molecular principles. We know a lot of the structures, but we do not know how those neurons are connected to give whatever the human intelligence we have, how this consciousness are building, we do not know, right? However, what we know, this brain is doing a computation. Sometimes we call this is the best computer in the universe or something. So a biological cell, okay, they do compute when they're connected in a proper way, they compute. And this computation is, you can imagine, this is probably, as I said, it's probably the best computer in the universe or something like that, okay? So it computes in a very complex ways, and this gives not only the intelligence, but the consciousness. So people want to try to understand that. And there is one project I like to mention, and this is called the Blue Brain Project. And this Blue Brain Project actually developed, so it is in the Ecole Polytechnique in Swiss institute along with the IBM. So their idea, so to develop a proper, develop a connectome of the brains the same way, you simulate in the computers in such a way that exactly the way the brain do it. So that means that you have the all connections, so you connect to simulate in a very realistic simulation of the brain for the rat brain. That was their main motto, but they want to go for the human, so that if you can do that. Still they haven't able to do that. They have a lot of they have done. That is not going to tell much of this, but still this is a very ambitious project to understand that how a mammalian brain function in that way. Now, interestingly, when you have this whole human brain simulation, still they haven't done that. But what I'm trying to, when they will use, what kind of the energy it will take. So if you want to do just a single human brain with all its connectome, if you want to do this in the computer, it will take 1 megawatt of the power. And 1 megawatt of the power means that much watt, and it means, if just want to tell you, this requires a mini hydropower generation plant to make that much of the power. Okay. However, when you think about the human brain, it takes only 20 watt. So compared to this 1 megawatt, just to see, right? There's a 6 zeros after 1, that watts of mini hydropower, compared to the human brain only the 20 watts, and do all kinds of this work. So if you see the computation in the biology, it is not only the superiority just in the number of computation they're doing, but at the same time, it's the energy budget. There's no match between 1 megawatt and this 20 watt, right? So there's a great. Now, okay, so that we all know about how human brain is doing this kind of computation. But the whole question is, can a small biological system like a bacteria, right, can they do some computation? And here I just want to give you some analogy with a movie. It's called Gajini. And in this Gajini movie, which is basically played by Amir Khan, and what they're trying to. So now there's I don't know if you have seen this movie. If you haven't seen this, not a problem. But just to tell you that how it's connected to this bacteria in one way. So in this movie, Amir Khan actually played a role where he forgets everything. Okay, and he had to take a revenge. That's why he wrote everything in his body, on his body, and then he cannot remember all the time, but he can read, and then he want to take that revenge in that way. In one scene, there is what happens. The Amir Khan was chasing a man, okay, a bad guy. So he was chasing, and suddenly what happens? There was a truck was coming from the other side. It hits that bad guy, and probably bad guy dies. Okay. Now at exactly that moment, Amir Khan forgets everything. So when Amir Khan forgets everything, and exactly at that moment, Amir Khan looks perplexed. Okay, he didn't know that what is happening, right? He couldn't remember. So what it is teaching us, this scene of this movie, is that if you want to do your next part of the work, then you have to remember the previous moments. What basically that means? That basically means that when Amir Khan was chasing that guy, he knows that I'm chasing, something like that, right? And at the moment, he forgets everything, okay? And he forgets everything. So he could not remember anything, so he cannot do anything in the future, right? That is the point. Now, if you think about your a term which you already actually learned, at least in your high school, and this chemotaxis, and this chemotaxis is the bacteria is so here this bacteria actually attracts towards some of the food particles, right? Some of the chemicals. So that is the chemotaxis. So now think about a thought experiment where you have a beaker, and in this beaker, at the one end of this beaker, you actually, this is the beaker with water, and you give some sugar, sucrose, a crystal, Okay, and this sugar crystal is now going to dissolve or diffuse so that you get a concentration gradient from one side of this beaker to the another side of the beaker. Okay, now now you put some bacteria on the other side of the beaker. Now what this bacteria will do, bacteria sends this sucrose and then it is going towards that towards the concentration gradient, the where concentration highs, towards the sugar crystals. Now when this bacteria is going through it, there's in the water there's some turbulence, and that turbulence actually hit the bacteria, and then bacteria actually just actually moves to the other way around. Now when it just goes to the other way around, now bacteria actually stops, because in the other way around, if it starts swimming, then it will actually go towards the other end of the sugar, right? So that it cannot get food and it cannot survive. So how this bacteria decides that when to stop and when to swim? In order to do that, definitely it has to calculate two things. That is, in its previous instance, it has to understand, the bacteria has to understand that what is the concentration of the sugar and the previous thing. And then on the next instant, it has to calculate the sugar concentration, and then it has to compare. If it compares the sugar concentration is high, then it will go, it is just swim. And if we found that it is not high, it is low, then it stops, right? So now this tells that it has to remember the previous instance. Now bacteria doesn't have brain, but bacteria does have memory. If bacteria doesn't have this memory, it cannot survive. So how this memory is coming? So this memory is coming through the interactions of the molecules. So a molecules can actually interact, come and develop a computation like a memory. Okay. So what to say? Okay. So exactly, if you want to develop this kind of the memory system, so how you are going to develop that? And that is the starting point of the synthetic biology. What people actually developed in 2000 is a Nature paper by Jim Collins group, and they developed something called genetic toggle switch, which is basically nothing but a simple memory device. So what they have to, so now this memory, so this is a switch. So now when you talk about the switch is the simplest memory device. Now there are two things. For example, let's say if this class won't be an online class, but if it is an offline class and I'm just showing some PowerPoints, I have a laser pointer in my hand, and if I press the laser pointer, then there is a light, right? A red or green light through which I'm just going through this presentation. Now, if I take my fingers off, there is no light. We call it switch, but it is not as a switch because it doesn't have a memory. It cannot remember that I pressed it. But if you think about the light switch in your room, so you actually press that light in the room. So, and then you take your fingers off, still it is on. Now, again, you turned it off, okay? Takes your fingers off, still it is off. So this is completely different than the laser pointer knob. So that actual mechanical switch for your light, that is a simplest memory device. So it remembers that someone pressed it and pressed it. And that is what you can actually see from here, what they have developed. So they developed a very simple plasmid. A plasmid is, and there's some genes and the promoters, and the way they actually developed, so what it will do, so if you give some small molecule, it's called IPTG, it is an inducer. So if you give to that system, that inducer, and then it is going to produce something called EGFP. It's a green fluorescence protein. So, and you can see from this figure, so if you give this IPTG, it is going to produce that EGFP. Now, this kind of system probably you have studied in your biochemistry book if you have a biochemistry, molecular biology, or any biology kind of the things, right? But what happens if you take the IPTG off from the system, you will see they are going to decrease. Okay, now they are going to decrease. So that is what you have actually generally learned in your biochemistry book. And what they have developed, that you give the IPTG, it is going up, and now you take the IPTG off from the system, and still the EGFP is turned on. EGFP is producing. Okay. Now you give the ATC into the system, it comes off, you take the ATC off from the system, still it is turned off. It is exactly like the mechanical switch or the light switch I have actually talked about. So in that way, you can actually see that it's the simplest memory device. Now, if you want to develop this kind of the memory device, how we are going to do that? First, you took a principle from the engineering, and it is called RS latch. It looks like that. You do not have to understand at this moment what RS latch is. So very first thing, if you want to, what is the manifestation of the RS latch in biological regime? Biological regime, it says that if you have two mutually repressing promoter gene pairs, it will give a similar kind of qualitative behavior. But this is a qualitative principle. If you know this qualitative principle, still you cannot make this memory device. Why you cannot make this memory device with a qualitative principle? Because it has a quantitative principle, and that quantitative principle says only at a certain parameter range it will give this bistable behavior or this memory kind of the behavior. If this parameter range out of this parameter range, it will give a monostable or like the laser pointer knob where it doesn't have the memory. So when you know this parameter range, then what you can do, then you can actually arrange your promoter and genes in such a way and brings their kinetic parameters by changing the translation rate, changing the interactions between the protein and genes in such a way it actually falls in this parameter range, and exactly the way they actually develop it, and that is why this gives a memory device. But if it is out of the parameter range, it will give a monostable behavior. So you give the IPDG, it shows the EGFP, you take the IPDG up, it is turned off because it is in the monostable regime. So now this is the starting point of the synthetic biology in that way. Now this is a difference between the biological, the biotechnology or the genetic engineering. Here, you first need a quantitative design principle, and based on that you actually develop your system so that you can actually make a basically toggle switch or the memory device. That's a very important point. That is a fundamental difference between these two. Okay, the biotechnology and the synthetic biology. So what you can do with this synthetic biology when you have this? So as I showed before, that the one goal of this synthetic biology that you want to use the electrical engineering principle where you actually connect different kind of biological gates and try to develop some complex functions similar to its computers. So people actually develop all the basic biological, all the logic gates using the biology. So what is this logic gates you are using? So the way you are doing, so if you think about an engineering a logic gate in computer, they're your on and off. These systems are basically flows of the electrons, or there is no flow of the electrons, right? Similar, but in biological system, you just abstract this idea. So your logic level 0, okay, is like a chemical. So presence of the inducer or not, or any other chemicals inside the cell is present or not. If it is present, it is 1. If it is not present, it is kind of 0. And it is going to produce some other biomolecules like proteins, like metabolites, and that's your output. And it's either it's on, either it is producing in a high amount, or it is producing in the 0 or the low amount. So that is the 0 and 1. So when you connect these things, then people actually develops a lot of the logic gates in the biology, all the basic logic gates people developed inside the cell and the synthetic biology. And then they also try to develop, if you see that what they have developed some more complex circuits. So here you can see people developed using the microRNA a circuit which is called a classifier circuit. So you connect those kind of the gates in such a way, developing some RNA gates. So what will happen? So if you take a HeLa cell, so HeLa cell has a particular microRNA signature, which is different than the other normal cell. So if you put your circuit inside this RNA circuit inside the HeLa cell, so this circuit will try to match its mRNA signature. And if they found there is a match between the mRNA signature in the HeLa cell and what is already been programmed, then it starts producing one protein, which actually helps this HeLa cell go to the apoptosis. Whereas if you put this classifier circuit in another normal cell, so you will see the microRNA signature of the normal cell is different than the HeLa cell, and it is not going to match. And what they will, if they don't found any match, this is not going to do anything. So the cell will just go out like that, right? So this is a proof of principle that when you think about this programming or this kind of things using this biology, you can do many things which you cannot do with the genetic engineering, the conventional biotechnology. So the whole question is, again, if I come to this, that why in biology we do not engineering principle and why this kind of the logic gates are required. So there's a one way to tell that. So if you think about your computer, what you're using at this moment. So this computer, they do have something called arithmetic logic unit. And this arithmetic logic unit has something called adder and subtractor. And that's where a computer actually add numbers, subtract numbers, or something like that, right? So this arithmetic logic units are the adders. So if you want to see this, what adder is nothing but something like that. It is a full adder truth table, and then it is the circuit. So if you can develop this circuit, you can make it full adder. So what people actually develop, the same kind of the adder biologically in a mammalian cell. So they follow the same kind of electronic principle. They connect them then together, and then what they got, the adder, where your output is some fluorescence protein, your input is some chemicals or hormones or something like that, right? Now the whole question is, if I tell someone that biologically, can you make this kind of function, okay, without knowing this engineering principle, just by thinking about the genes and the proteins together? You cannot come up with a network, a biological or gene regulatory network, which will give you this kind of behavior just by thinking, okay? So why engineering is important? Exactly here engineering is important, because we know from this engineering that if you have some of the logic gates, the basic logic gates, and if you want to have any function, you can just add according to this engineering design principle, the same way as an adder, and it will give you the actual behavior. So that's why they actually develop. They don't have to know that how this particular protein is going to connect with that, how this is going to regulate, how that regulation is going to help it, this particular things, and how this is going to that. You don't need to know. You just connect them together and to actually develop the whole whole function. So that is why the engineering principle is important, which you cannot develop just by thinking, but when you use the engineering principle, it would be very, very easy for you. I mean, it is not easy practically, because when you're doing the biological system, but what I'm trying to say, that it is theoretically, it is easy, because you don't have to think how these things are inside, how they're actually interacting with each other. So that is why, so you can develop any function, because through this electronics, I mean, whatever a computer is, it is nothing but the gates, but this computer have all things are doing for you. So you can develop lots of function if you actually use this kind of engineering principle in the biology. And that is the motto of the synthetic biology, through which you can actually develop something like that. Now, even if you do, so what could be its application of this kind of the system? And even if you take very simple device, then you can also see there's a lot of different applications. And one is that you can think about your therapeutics, what you know today, as a program therapeutics. What is the program therapeutics? For example, here I give an example where the people engineered the E. coli, okay? E. coli cell in such a way that says E. coli is a gut bacteria, you know, that's some species of the E. coli is a gut bacteria. And you engineered the E. coli in such a way that when there are some pathogens are coming, okay, and then these pathogens are always producing some of the chemicals, okay, and that chemicals can be sensed by this engineered E. coli. And when they sense that, okay, this particular pathogen is present, they start producing a specific kind of peptides which will only kill that pathogen, not the others. So you can see it is completely, it's much better in terms, I mean, compared to the antibiotics, if it is really comes into the market, because the antibiotics, when you use the antibiotics, it kills all other bacteria. But here it is very specific, okay? And this, your engineered gut bacteria or E. coli, it will only turn off or turned on when there is some other pathogens are coming and secreting some chemicals. So it is a completely programmed. Okay, so similarly, people actually developed something in the mouse where they engineered the cell in such a way. You know, there's the uric acid. So many human has a problem of having a lot of uric acid in his body or her body. And so here they actually developed some uric acid homeostasis, some cellular engineering for uric acid homeostasis. So this engineered cell you inject in the mouse, and in this mouse, if there is any increase in the uric acid level, then this engineered cell can sense that and then produce some enzymes to actually reduce that particular uric acid to turning off some kind of the pathways. Okay, and so that's, you can see this. Then again, this is clearly is a program therapy. It is programmed. It is not the passive medicine you are giving someone, but your engineered cell can actually sense that the system, and then it decides that what to do. Okay, so this is the kind of level of application you could think about the synthetic biology. So the next is a very interesting applications where you used a bacterial lawn, like in an agar plate, and you use this as a negative. So today is like a digital thing, but digital photographs. But like 20 years back, the photographs were basically you have to print them, right? You have to print on a negative. First, you have a negative in the film, and then you can, from the negative, you actually develop that. So here it is exactly like those kind of the bacteria. So how they actually develop? So they developed a bacteria, and this engineered in such a way, if there is a little more light comes, it gives a darker, and if it is less light come, it gives a lighter shade on the bacteria. And how they, by producing some chemicals which basically black, or the other way around, you can also do that. So on that bacterial film, you take a photograph and or negative, and then you project it here. And when you project it, so because based on this negative, there would be different light shades will come on this bacterial lawn, and based on that, it will start producing that black chemicals, and you basically actually develop a printing. So you can actually make the photographs using engineered bacteria. So this is a very fun application, but this is just going that what is the potential of the synthetic biology you can think about today. So here is another example where the idea of the scientist that you develop a plant. Okay, so if there is an environmental pollutants, or if there is a explosive like the RDX or TNT, then this the plant, the engineered plant, it can actually sense that, and when they sense that, it turns white from the green, and people actually develop that. And what they took some Arabidopsis, and they engineered in such a way that if you give the TNT, okay, trinitrotoluene, that's an used in several explosives. So if these explosives are there in the environment, so they can sense, and when they sense, this they become white. How they become white? Because the sensor is connected with the chlorophyll making mechanism. So if this sense that TNT, there is they engineered the gene regulatory system in such a way that this basically the chlorophyll producing mechanism will be stopped, and it comes actually as a white things. Okay, so this is again now. This won't be used as a bomb detecting plant because it takes like more than 24 hours. Okay, so you cannot. But okay, people are trying to make it as faster. But if you don't think as explosive, but think about the environmental pollutants. So like if you have some tree at your home or at your laboratory or to your office, and then if there is some pollutants are coming to your area, then this kind of plant can actually sense that, and changing its color will tell there is some kind of the pollutants or very specific pollutants is actually building up in your area. So in that way, this is a very nice applications on that. Now, there is another part of synthetic biology. So synthetic biology has, as I said, it is started with as a set of the engineers who wants to develop the biological engineering exactly like in the other engineering, like mechanical engineering, the computer engineering. But there is another set of people who try to develop something which is called synthetic metabolic engineering. So metabolic engineering is a very old field where you engineer the metabolic network of a bacteria by overexpressing some genes or deleting or mutating some things, and then you actually trying to produce a particular chemicals which has already been produced by this bacteria or the yeast to actually increase its flux, so that you can increase something like the antibiotic. many many bacteria produce antibiotics. So how to increase the amount of the antibiotics, or many secondary chemicals that produce, and how to increase that particular secondary metabolites for industrial application. That's the metabolic engineering. Now, what this synthetic metabolic engineering tells, okay, so now you can actually change the flux of something in your organism. Then why not you bring the other metabolic pathway from other organism and put in the bacteria or yeast? For example, one of the success story is production of the artemisinin. So this artemisinin is a drug for the malaria. And you probably know a few years back, one Chinese lady who actually got the Nobel Prize for discovering artemisinin as a medicine for malaria. But this artemisinin is very, very costly because they produce from the drug called Artemisia annua. And this, means you have to extract that artemisinin from here. So extracting from plant, you can imagine there's a very costly business. Okay, so artemisinin is a very costly drug. So synthetic biologists thought that why not you take this artemisinin production pathway and put it in the bacteria or the yeast, so that you can produce this artemisinin in a reactor, not on a field. So exactly they have done in the both E. coli and the yeast, and that is a success because it's a commercial success. Now they developed in the reactor, and now artemisinin they actually sell, right? So that is a very interesting story on that. Now, but this gives a lot of potential. What potential? Now think about, you probably know that all what we get the drugs today, let's say the cancer drugs, most of them are coming from the plant sources, okay? And many of the cases you cannot chemically synthesize that, right? So to do that, many people actually looking at getting some new kinds of the chemicals, new kind of the drugs, and everything from the plant. So that is, there's a lot of fishing experiments are going on based on the Amazon rainforest. So they take a lot of different kind of plants from there, they extract it and then use in many ways. So one way to actually you think about that, if you really find a plant from the Amazon rainforest, and which is very important for the whole world, then it is very difficult to actually cut all the trees from the Amazon, okay? Because that will give us a huge problem to our climates and many things, okay? So the whole idea of the synthetic biology, if you can take that pathway and actually put in the yeast or the bacteria, then you can actually produce those kind of the chemicals in the yeast and the bacteria, okay? So that you can imagine that's what kind of the potential application of this synthetic metabolic engineering can give you. A similar other things you think about for the biofuel. Now you know that there is a lot of controversies when you see any paper nowadays in either New York Times or any science MIT Technological Review and some or any business Wall Street journals, because there is a controversy because now people grow corns for basically for the biofuels, and they take the huge amount of the field. So the question is, so in later you're going to use the corn for the food, or you're going to use for the fuel. Okay, so that's this kind of controversy. So think about, instead of making this plant, you are actually going to produce your biofuel. Either it's an diesel, or sometimes people think about the hydrogen on cyanobacteria. And then when you think about the cyanobacterial algae, you don't need basically this kind of the field, but you need some of the ponds where you actually put this kind of cyanobacteria, which is engineered, and is engineered in such a way that the biodiesel, which is going to be produced from this kind of from the corns, those kind of things, you develop some of the synthetic pathway inside the bacteria or the cyanobacteria or the algae, which is going to give you those kind of the biofuels, right? So so you can see that is the, again, another potential. Many people are working on that. So that is one part. There's a very interesting company, it's called Intruxon. The Intruxon is a very petrochemical company, and the biotechnology company basically. Now they're going to the synthetic biology. And so you know the way the petrochemicals people developed, they take the fossil fuels from the earth, and that is a long carbon chains, and then you actually divide these carbon chains to ethylene, and then you start making polythene, polypropylene, and so on, your petrol, your diesel, your kerosene, your aviation fuel, and everything. So this people thinks, no, not that other way, but you can start from a natural gas. You take the natural gas or the something carbon or the methane or something, or even carbon dioxide, and you start adding those carbon together to make those kind of ethylene, polypropylene, etc. Instead of dividing those carbon chains, you start adding those carbon chains to do that. So they use this synthetic biology. They don't share that what is the mechanism of the synthetic biology they're using, but definitely it's a synthetic metabolic engineering, and that way they want to actually develop fuels, they want to develop chemicals, the proteins completely from this bottom up. So that's an one success story of the synthetic biology people are thinking. Now, so today is again, it's an introductory lecture. I'm trying to give that where is this potential, and from the next class we may go a little bit deeper how we are going to develop that. But here one thing is very important. So this is a very inspiring story. It is nothing about the synthetic biology, but it is a very inspiring story. Her name is Alexandria, and so she had a problem of heart failure. Okay, so because she started her life with a defective heart, and it is a time when it is in 2020, last year. Okay, and that is a corona everywhere. So it's a New York, in a New York hospital, the doctors actually replaced her heart with some other heart. Okay, and most of the cases, this kind of when this is just a 9-month-old child, most of the cases child died. Okay, they could not take that kind of operation, or most of the time you cannot get a proper heart for, because the heart replacement, you know, say you need another person who is in the coma or something, and you cannot have a person as a, some old person, because this is just a 9 months, so his heart has to match. But somehow it got, and even in this corona thing, everybody thought she could not survive, but she survived. So it is a very inspiring story. However, when this story is inspiring, means, as I said, there is some other dark stories, right, where those children could not survive. So only solution for this kind of congenital heart defects of the child, the only solution is if you can develop a heart in vitro. Okay, and exactly heart from probably the patient's own cell, and then it will give you these things. And that comes from a perspective of the tissue engineering. Probably you heard of the tissue engineering. The whole idea of the tissue engineering, you develop some of the organs from outside your body in a reactor or in a lab. So what we know today, that from a stem cell, even from the skin cell, you can actually develop some of the heart cells. But when you're developing the heart cells is not going to make the heart, because heart is a very complex organ with a lot of different kind of complex functions. So how we are going to develop that? The tissue engineer, the way they try to do that, because it requires a 3-dimensional functional structure. So the tissue engineers, what they do, they use the materials, different kind of the material scaffold, and then they actually embed those cells, and those, when these cells are growing, they are taking the shape of this 3D object. And so what here is one success story. Here you can see it is a bladder for the pig, the urinary bladder of the pig, and basically they use this material scaffold. There's a spherical material scaffold. They put this kind of pig cells into it, and they actually develops as a urinary bladder of the pig. But this is, you can see, this spherical, a simple object, though 3D, but still it's a simple object. This kind of making this kind of object materialistic and functionally, it is completely impossible at this moment. Okay. Now, synthetic biology wants to actually intervene here and think from a different perspective. Now, believe all of our life has been started from a single cell, right? And then when we are growing in mother's womb, what we found that all of our organs are growing without any material scaffold, right? So that there is something is already been written in our genome, which has the information to how to make this complex heart or a kidney or something, this complex design, functional design. So the synthetic biologists thought that if we somehow tap or hack those kind of the function, so probably without any material scaffold, we can actually develop this kind of, I mean, we don't know with material scaffold we can make a heart, but at least if we can actually hack the information from this, then probably one day we'll be able to make this kind of the very functional very functional heart or something. So to go that, we have to start something from a very old physical chemistry, and it is called the BZ reactions, okay? And it's called the start. So if you take this just 1, 2, 3, 4 chemicals at certain concentration, and you mix it into the beaker, and you just stir it, and if you stay, what you'll see that first is give a purple color, and with time, this purple color is going to be changed to red. That's good. Sorry, red, I mean blue. And this blue again going to be changed as the purple. And again, this purple going to be go to the blue. So the reaction is oscillating, but this is a very important reaction because you have not seen, I mean, whatever the chemistry we studied, whatever the practical or experiments we do in the chemical lab, there you mix the chemicals, and these chemicals at a certain point goes to the equilibrium, and if there's a change of the color is changed and then stop there. If you think about the acid-base titration, if it's going to from a colorless to the blue or the or red, and it stays there. It is not coming again in the colorless, again it's turning blue. It is not happening by together. But this reaction is somehow, it's telling that it is going to actually oscillating. So it is out of the equilibrium for some time. So that's a very important point, because the same reaction, if you want to do without stirring, just put those chemicals, put in a beaker or a petri dish or something, with time, and you don't stir it. And with time, what you start seeing, that you can see a start of a pattern, a different kind of patterns. And that is, and these patterns is somehow connected directly to the growth of the heart. And we'll see in some next slides, and what are those. And so to do that, we need some person, and this is the guy. He is the father of the computer science. Okay, so what he says is Alan Turing, right? So what Alan Turing is not only the father of the computer science, but what he developed a theory in 1952, which is something called the reaction diffusions theory. And what, so you know that when something is developing, it is the developing Drosophila, and this Drosophila, when it's developing, it has some segmentation. So the question is, how in developmental biology this segmentation happens? The Alan Turing said that this kind of the segmentation is coming, this pattern kind of formation is coming from something called reaction diffusion system. So if you have some chemicals and they're part of them are reacting, and one of the product is diffusing at a certain manner, then you can actually mathematically develop some kind of the pattern, something like that. And exactly when people today see how this gene is patterning during the segmentation, you can find this kind of the pattern. So this pattern formation is a very important aspect of the developmental biology. Okay, and either developing heart, kidney, and will come in a moment. So this pattern formation, you have to bring it in the synthetic biology. So I'll take another 5 minutes probably, and then I'll stop today here, and then we'll start again from the next class. So now here, okay, here is I mean, how this is happening. If you want to see a bit, probably today I'm not going to talk about these slides because I have to explain a lot, because I'm not going to the mechanistic details today. Today is just an introduction. I'll discuss it in some other classes. But what I'll give an example, people actually found what is the reaction diffusion mechanism. For example, is a digit formation. So digit formation, we have our hand have these 5 fingers, but you see when my hand was developing, somehow this cell has to develop, this cell has to stop, this cell has to develop, this cell has to stop, this has to develop, this has to stop. So how this asymmetry is forming, people do know exactly in case of the chicken that how it is happening, just following this kind of few genes. Which genes are interacting, their products is diffusing, and there's a feedback. So if you find that, people now know that how this digit is forming based on this reaction diffusion system. So this proves that if you take those kind of the physical principle and actually put into the action, this is happening. So if you really put it in synthetically, then probably you can develop some of the patterns. And that's why here is the example here. So here, what you find that in 2005, Ronway's group, actually, this is nothing but bacteria. The lawn of the bacteria on a petri dish, and then they're engineered in such a way, so they produce 2 protein, one is red fluorescence protein, and is green fluorescence protein. But when they're producing this kind of the protein, you are not saying how to produce, but you just give some inducer and they are forming their own pattern. Okay, it is just a cell, you just put an inducer and they start producing this kind of the pattern. And now very interestingly, among these 5, one of them is the computer simulation of the system and other 4 are basically an experiment. Now just looking at it, you cannot tell that which one is the computer simulation and which one is the experiment. In reality, actually, I also forgot that which one is what. Like if you go to this paper, you can easily see that which one when they said that. So basically say that you can develop this kind of the pattern forming exactly using the engineering synthetic biology principle and put it in the bacteria. So this is 2005, and it is probably, this one is probably in 2000, somehow it didn't come, which I wrote, but this is probably 2015. They actually developed this kind of the concentric. So here you can see only one. I mean, this is the inducer. So there's so 1, 2. So there's only 2 concentric cycles. Here you see there's the 5, 6 different concentric cycles. This is similar like this Drosophila, where you can see this kind of just pattern formation. And this pattern formation is again not in the 3-dimensional, but it is in the 2-dimensional. But what I'm trying to tell you that synthetic biology is now going to exactly in the design principle and then engineer in such a way that you can actually produce this kind of the pattern. People develop this kind of the pattern again in the mammalian cell. This is a 3D mammalian cell things, but here the green and red, it is programmed again. So this is not just an anything, but they are programmed in such a way that you can develop this kind of the patterns. So this said that you can generate some 3D patterning using the synthetic circuits. So we believe probably, I don't know, next 20, 25 years or 50 years, probably you are the one of them who can actually really develop a functional thing, because you started with the exact principle of what a developmental biology follows. So for today, I'll actually stop here. So if you have any questions, you can write in the chat box, or I can also discuss those things the next week. And I'll stop here. I'll give it to Professor Partho Saha.
Sangram.
Yes.
Actually, that time I tried to log in, but there's some technical issues, I guess.
Yeah. Actually, yeah, I was also told that something was going on there.
So
yeah.
I think everything went fine, right?
Yes, everything went fine, seems.
Oh. Okay, fine, Sangram. So today's lecture is over, right?
Yeah, today's lecture is over. Yes.
Okay.
So my next lecture in the Monday. So if you don't have a question, so I think I can stop with the permission.
So there is no more questions, sir.
Okay, so there's no more question. So I think.
So we'll stop the session, right?
Okay. Okay. Thank you very much. Thank you very much.